Just as a reminder of the overall reaction in respiration, you start with glucose and it overall reacts with oxygen. And when you do that, you release energy in the form of ATP, and the glucose is broken down into carbon dioxide and water. So for one glucose molecule, you use up 6 oxygens, and you get back 6 CO2s and 6 molecules of water. Now this slide is actually found in your book where it goes through the reactions of glycolysis step by step. So this is a reaction sequence for glycolysis starting with glucose and ending up with pyruvate. You can, and I recommend that you look at this in your book and read everything that happens step by step. I'm not going to be using it in my presentation. I'm going to use the picture that I supplied with your notes, and I'll skip ahead to that now. This slide shows you the overall scheme for glycolysis, starting with glucose and ending down at pyruvate, and then we'll also cover fermentation at the bottom of that slide, and we'll do it step by step. Keep in mind that, first of all, glycolysis is broken up into 2 sequences of steps. The first is called the preparatory steps, where you are sort of gearing up for making ATP. You're actually adding energy to the process. And then after that are the steps where you actually make ATP. Also, notice that in this particular sequence of reactions, I follow the bouncing carbons. And notice that glucose is denoted as a C6 molecule, and some of the others are denoted as C3 molecules. And I'm just interested in where the carbons are and not so much the extraneous stuff. Okay, we'll start here. I've added some things to this slide. Notice the preparatory steps are at the top, and then the oxidation steps that will produce ATP are at the bottom. And we'll keep, while we're doing this, an ATP tally sheet in red on the right-hand side of the slide as we move down. We start glycolysis with glucose, a 6-carbon molecule, and given the fact that we intend to make ATP here, it may seem kind of strange that we start off by using up an ATP. We take the glucose molecule, ATP is used up, and the phosphate group that's released is attached to glucose. So instead of having gained ATPs, we're actually starting off here down 1 ATP, a -1 in ATP. Now, if that wasn't good enough, what do we do next? But we add another phosphate group. We break down another ATP molecule, and now instead of being down 1 ATP, we're actually down 2 ATPs, and we end up with a molecule that has a phosphate group on either side. It's a 6-carbon molecule with a phosphate group on one end and a phosphate group on the other end, and we've used up 2 ATPs. Now, you may or may not have an investment counselor at this point, but one thing that your investment counselor will always tell you is that in order to make money, you have to spend money. And that's effectively what's happened here. We've spent ATPs, 2 of them to be exact, in order to make more ATP than we use up. We're going to have a net gain at the end of this process. Now, it turns out that the 6-carbon molecule with 2 phosphates on either end that was made after using up those 2 ATPs, turns out that that molecule is very unstable, and it will break down immediately into 2 3-carbon fragments, each of which has a phosphate group on one end. This molecule is called phosphoglyceraldehyde, or PGAL, and this breakdown again is instantaneous. Also, this marks the end of the preparatory or pump priming stages of glycolysis. From here on out, we're going to be making ATPs. Now, what you need to keep in mind is that we started with one glucose molecule that had 6 carbons, and we have to keep track of all 6 of those carbons. So for the rest of this process, we're going to keep track of both of the PGAL molecules that were made during the pump priming stages of glycolysis. So now we have to follow 2 3-carbon fragments through this process. Now we'll start the oxidation portion of glycolysis that ends up making ATP. The first thing we do here is NAD comes in, and one NAD molecule, one oxidized NAD molecule will act on each of the 2,3 carbon fragments that we brought in. It'll come in and it'll steal some electrons from PGAL. And those electrons will be bound to NAD to make reduced NAD. So you're going to bind electrons to oxidized NAD to make reduced NAD. And whenever you run one of these reactions, one molecule is oxidized and the other one is reduced. In this particular case, NAD is reduced and the PGAL molecules are oxidized by the removal of the electrons. Now, when that happens, it makes these 2,3 carbon fragments with one phosphate group unstable. And they relieve this instability by picking up phosphates from the surrounding solution, the surrounding milieu in the cytoplasm. Remember, of course, that glycolysis, and I didn't mention this before, but glycolysis occurs in the cytoplasm of the cell, outside the mitochondria. So the phosphate group comes on, and notice that when the phosphate groups bind to each of these 2,3 carbon fragments, it binds by a high energy bond. You can see that wavy bond there. And so now we have these two high energy phosphate groups bound there, and interestingly, the energy with which they are bound to PGAL is higher than the energy required to make ATP. So that when this bond is broken, more energy is released than is required to make ATP from ADP in a phosphate group. So this will then be a coupled reaction where the when breaking off this phosphate group releases more energy than is needed to produce ATP from ADP and phosphate. The result is that's a negative delta G reaction. It's actually two coupled reactions, the breakdown of this 3 carbon fragment with 2 phosphates on it, and the production of ATP from ADP in a phosphate group. Now in this next step, that high energy phosphate on the 2,3 carbon fragments with the 2 phosphate groups on it, the high energy phosphate is broken off and that phosphate group is added to 2 ADP molecules to make 2 ATP molecules. And what we end up with is we got 2 ATPs produced here. We had used up 2 before. So now our energy balance sheet, our ATP balance sheet, tally sheet, takes us to 0. We have now broken even on the number of ATPs. We used 2 up in the preparatory steps. We got those 2 back now. Now the 3 carbon molecule that we produced, it looks the same on this drawing as PGAL, but it is not PGAL. It is a different molecule, but I just wanted to point out to you that even though it looks the same on the drawing, it is in fact a different molecule. Now in the next step in this process, what will happen is the 3 carbon molecules with the phosphate group on them, the 2 3 carbon molecules with the phosphate group on them, they are acted on by an enzyme, and that enzyme will remove water from this molecule. The removal of water from these 2 molecules will change them and destabilize them in such a way that the structure changes so that the phosphate group, which was originally attached by a regular low energy bond, that bond energy is increased so that once again we have a high energy bond, and once again that high energy bond contains more energy than is needed to, when that phosphate group breaks off, to make ATP. So once again, as you're going to see in the next step, that bond will be broken, the phosphate group will be added to ADP to make ATP, and once again it'll be a coupled reaction with this particular reaction that we're going to see in a moment, giving off more energy than is needed to make ATP, coupled reactions, overall negative delta G, and then there you have it. There we will make another ATP. Actually, 2 more ATPs. Okay, here you see what I was talking about a moment ago happening. The high energy phosphate breaks off. It is transferred to ADP to make 2 ATPs, 1 from each of those 2 3 carbon molecules. And if you look over at the tally sheet where we had broken even a couple steps before, now we are up 2 ATPs. We have now, for that glucose molecule, we have produced 2 ATPs. And the end product of this, the product at the bottom of glycolysis, is a molecule called pyruvate. It is a 3 carbon molecule, and you can see it there at the bottom of the sequence. And we've made 2 of them because we, again, keep in mind, we have to follow all 6 carbons that came from the original glucose molecule that started this process.